everyone. Today we are going to learn electrochemical nanobiosensor. In today's lecture, we are going to learn what is a biosensor, what are the various components of biosensor, and we are also going to learn how does electrochemical biosensor work both theoretically as well as practically through demonstration. Let us see what is a biosensor. A biosensor is a sensor that integrates a biological element with a physiological transducer to produce an electronic signal proportional to an analyte concentration which is conveyed to a detector. In today's lecture, we are going to learn all these in detail in the subsequent slides. Let us briefly learn the history of biosensor. Dr. Clark was the first one who introduced the concept of biosensor, in which he incorporated an enzyme as a bioreceptor to an electrode where he can sense the analyte based on electrochemical change. Originally, it was designed to detect oxygen, which we also called as Clark electrode. With furthermore development, he has also developed for glucose sensing, and based on this pioneering work, now we can see a most successful commercialized product, that is glucose biosensor. In this picture, you can see that the glucose oxidase enzyme has been immobilized on an electrode surface, and when the glucose analyte comes in contact, reaction takes place on the anodic as well as the cathodic electrodes, which generate an electrochemical change to detect the glucose. Let us see the development of biosensor. In 1962, the first glucose biosensor was developed. Followed by that, you can see that the immunosensor was developed in 1985. In 2000, wearable and implantable biosensors are developed, and from 2010 onwards, you can see the role of nanotechnology in biosensor. From this slide, you can clearly understand the development of biosensor, starting from oxygen electrode that was developed in 1956, followed by glucose biosensor in 1962, which is one of the successful biosensor till date. And there are a lot of development happens in this field, especially it was started in 1975 where the immunosensor was developed, and 1998 DNA based biosensor, and presently you can see here there are a lot of sensor like a contact lens biosensor, calorimetric sweat biosensor, and flexible and wearable biosensor, and presently we are using these AI and ML based biosensor. Let us see the characteristics of nanobiosensor. The first one is sensitivity, that means it detects small changes in analyte concentration, and next one is specificity, that means its ability to detect a specific analyte distinguishes target analyte from similar, and the third one is linearity, that is the direct proportionality between response and concentration, and fourth one is the response time, that means the rapid time to reach a stable signal, and fifth one is the reproducibility, that is the consistency of results across tests, and the last one is the dynamic range, that means the range of reliable analyte concentrations. Let us learn about the various components of biosensor. The first one is the target analyte, which represents the substance of interest, and the bioreceptor, so is a specialized molecule or system designed to specifically recognize and bind to the target analyte. So, you can see here, for example, it can be a antibody specific for the particular antigen. So, this can come and bind to the bioreceptor. This analyte can come and bind to the bioreceptor, and this bioreceptor is immobilized onto the transducer, which is responsible for detecting and converting the biological interaction into a measurable signal. So, once there is an antigen antibody complex, it will measure the signal, and this signal is subsequently amplified by the amplifier, and the signal processor then refines and quantifies the amplified signal, which is finally displayed as a measurable output on the display unit. In the following slides, we will go deeper into each of these components to better understand their roles and functions. Let us see the first component of biosensor, that is target analyte. Malfunction at the cellular level triggers the release of specific molecular markers, which serve as early indicators of disease. As the condition advances, these biomarkers accumulate and their concentration rise significantly. So, therefore, early detection of these biomarker is critical to ensure timely diagnosis and intervention. As I told earlier, any disease, it starts at the molecular level. In the molecular level, you get the signals in femtomolar or in the picomolar or nanomolar. To detect this signals, we have to use a very sensitive biosensor. With the progress in the disease, what happens is the biomarkers also accumulate, there will be a rise in the biomarker. But if you are able to detect the disease in the later stage at the organ level or in the system level, it is very difficult to cure the disease. But if you are able to detect the disease at the early stage at the molecular level using this sensitive biosensor, then we can overcome these diseases. The next component is bioreceptor. A bioreceptor specifically engages with the target analyte, and the bioreceptor ensures the sensor's selectivity and specificity by binding or reacting with the analyte through biological recognition mechanism. So, these are the various bioreceptors. The first one is enzyme based. So, here we can use the enzyme as a bioreceptor. Example is glucose oxidase for glucose sensing, and next one is we can use the antibody. These antibodies are highly specific for the particular biomarker. For example, if you are using the antibody specific for the lung cancer, it will detect the lung cancer, and if you are using the antibody specifically for the breast cancer, and it will specifically detect the breast cancer. The next one is we can also use the DNA or RNA as a kind of bioreceptor. Here the DNA or RNA will bind to the complementary sequence of the target nucleic acid, that is the DNA probes, which can be useful for genetic testing as well as the pathogen detection. For example, if there is a viral infection or bacterial infection, and if you do not have antibody for specifically for that particular virus or bacteria, we can use this DNA or RNA. And most of you know the DNA work based on the complementary sequences, and if we have the complementary sequences, it will bind and it will produce a signal, and that can be measured using this nucleic acid based sensor. And the fourth one is aptamer. This aptamer is a synthetic oligonucleotides or peptides, and that binds specifically to the target like proteins or small molecules. So, we can use this aptamer based sensor mainly for identifying these small drug molecules. The next one is the immobilized cells or tissue, that we can immobilize the living cells that can detect the specific environmental changes such as toxins or biological substances, and that can be detected by using this whole cell biosensor by immobilizing this whole cell. The next component is transducer. Transducer is one of the most crucial component of the biosensor, which transforms the biological interaction between the analyte and bioreceptor into a measurable signal. And we have several types of transducer. The first one is electrochemical transducer. It measures the changes in electric properties such as current, voltage or impedance caused by the biochemical reaction. And next one is optical transducer. It measures the changes in light properties like intensity, wavelength or phase or the polarization of the light due to an analyte and receptor interaction. So, when there is an interaction, for example, if you are getting a fluorescent signal, it can be fluorescent on or it can be fluorescent off. So, it can quantify and it can detect the particular analyte. The third one is acoustic transducer. It detects the changes in sound wave properties such as frequency and amplitude. And fourth one is piezoelectric transducer. It measures the changes in mechanical properties such as mass or resonance frequency caused by the interaction. And the last one is thermal transducer. It detects the temperature changes in terms of heat generated during the biochemical reaction. So, if there is a heat is generated, that can be detected using this thermal transducer, and if there is a mass or resonance frequency change, the mass or resonance frequency, that can be detected using this piezoelectric transducer. From this slide, you can clearly understand some of the examples of transducer as well as the application. So, under the electrochemical transducer, the examples are like amperometric, potentiometric and impedimetric. So, applications like we can use it for glucose sensor or pH meters, and under the piezoelectric transducer, we can have the example like QCM, and applications we can use it for gas sensor or pathogen detection. Under the acoustic transducer, we have the examples such as SAW sensor, which can be useful for environmental monitoring and food safety, and optical transducer examples are like fluorescence and SPR, that is a surface plasmon resonance. The applications like we can use it for DNA sensors and biomarker detection. So, the last one is the thermal transducer, and the example is calorimetric biosensor, and the applications is like we can use it for metabolic studies as well as the food testing. Let us see the classification of biosensor. The biosensor can be classified based on bioreceptor. It can be enzyme based or antibody based biosensor, or aptamer or whole cell and DNA based, which I already explained. And it can be based on detection technology. It can be based on nanomaterial based biosensor or SPR biosensor or magnetic biosensor. And it can be based on the transducer, that is like it can be optical, thermal or acoustic or gravimetric. And again, based on the modern development, we can use the advanced materials and we can reduce the size, we can have a small size and wearable devices, and we can also integrate the IoT, AI and ML to advance this biosensor. Let us see how does electrochemical biosensor work. Electrochemical biosensing is a study of a relationship between voltage, resistance or capacitance change and the identifiable chemical or biological change upon the analyte interaction. That means, once the analyte interact with the particular receptor and there will be changes. So, that can be capacitance or that can be voltage or it can be resistance. The capacitance change means it demonstrates the changes in stored electrical charge with analyte presence, and in case of resistance change, it reflects the alterations in the material conductivity upon analyte binding, and voltage change indicates a shift in electrical potential due to analyte interaction. Let us learn about the fundamentals of electrochemical biosensor. The foundation of electrochemical biosensor lies in redox reactions. According to redox nomenclature, a pair of species involved in electron transfer is referred to as a redox couple. Redox reactions in which one chemical species obtains electrons and another gives up electrons are the basic building blocks of electrochemical biosensor. In this picture, you can see here the target analyte undergoes diffusion to reach the biorecognition element, which is immobilized on the biosensor surface. The biorecognition element specifically interacts with the target analyte, triggering a change in the redox reaction. This reaction generates a flow of electrons, which is then captured and translated into an electrical signal by the transducer. And this magnitude of this signal is directly proportional to the concentration of the analyte as shown in this graph. The behavior of this redox reaction and the resulting signal can be mathematically described using the Nernst equation, and this equation explains how the potential generated during the redox process depends on the concentration of the redox species and other system parameters. By monitoring this potential or the resulting current, we can quantitatively detect the target analyte. So, this is the fundamental of electrochemical biosensor. Let us now explore the electrochemical workstation and the key components of 3 electrodes system, which is commonly used in biosensor application. And electrochemical workstation is a sophisticated instrument designed to control and measure the electrochemical reactions in a precise and controlled environment. So, it comprises of several components including potentiostat, which regulates the voltage between electrodes, and this potentiostat governs the interaction between these electrodes by applying a controlled voltage and measuring the resulting current. And this dynamic interplay provides critical information about the concentration and nature of analyte. And we have a computer interface to monitor and analyze the resulting data. And at the heart of this system, we have this 3 electrode setup, often integrated into a screen printed chip or electrode for compactness and ease of use. And it consists of working electrode, counter electrode and reference electrode. Working electrode is the electrode where the electrochemical reaction of interest occurs. It interacts with the analyte and measures the resulting current or potential changes. The next one is the counter electrode, also known as the auxiliary electrode. It completes the electrical circuit by allowing current to flow between itself and the working electrode. And third one is the reference electrode. So, this is an electrode that can maintain a constant potential under changing experimental conditions, and it is used as a reference point to measure and control the potential of the working electrode. Let us learn the role of cyclic voltammetry in biosensing. Cyclic voltammetry in biosensing is an electrochemical technique used to analyze the redox behavior of biomolecules and detect biological targets. This cyclic voltammetry involves applying a cyclic potential sweep to the working electrode while monitoring the resulting current. The applied potential is varied linearly with time in a triangular waveform as shown in this graph, and during the scan the potential causes oxidation and reduction of the analyte at the electrode surface, and these electrode reactions generate current responses that are recorded to produce a characteristic plot known as cyclic voltammogram. This cyclic voltammogram is a plot of current versus potential, providing vital information about the electrochemical properties of the analyte. And here we have this anodic peak corresponding to the oxidation process, that means loss of electrons, and we have the cathodic peak corresponding to the reduction process, that is a gain of electrons. The separation between the anodic and cathodic peaks provides insights into the reaction kinetics, while the peak intensities are directly related to the analyte concentration. In biosensing, CV is often used to study the interaction between the target analyte and the biosensor's biorecognition elements such as antibodies or enzymes. For example, the schematic here demonstrates the redox reaction of ferrocyanide and ferricyanate, which is frequently used as an electrochemical probe, and it is also called as a redox couple. And when a target analyte interacts with the biorecognition layer, it modulates the redox behavior of the probe and causing measurable changes in the voltammogram. Let us learn about how to fabricate immunoelectrode for biosensing application. So, it has 3 steps. The first one is electrode modification. So, we will be using the screen printed electrode, and the working electrode of the screen printed electrode will be modified using a nanomaterial. So, here we are going to use the functionalized graphene, and that will improve the conductivity and surface area. And you can deposit the functionalized graphene by drop casting method. Once the working electrode is modified, you can see that the CV curves shows an increase in the peak current, indicating the enhanced electron transfer. The next step is functionalization with antibodies. So, now we have to conjugate the antibody on the working electrode surface using a coupling agent, that is EDC NHS, and we are going to add the anti-CEA antibody, that is the antibody against carcinoembryonic antigen, and this will be immobilized on the graphene surface. And after this, you can see that the CV result shows a slight shift, so indicating the successful conjugation of the antibodies. The third step is blocking with BSA, that is bovine serum albumin. In this step, non-specific binding sites are blocked using BSA, and the CV response here ensures that the electrode remains selective for the target analyte and minimize the background noise. By blocking with BSA, we can prevent the non-specific binding and we can improve the efficiency of this biosensor. So, let us learn about how to interpret the CV results from this graph. And whenever we fabricate immunoelectrode for biosensing application, each and every step has to be confirmed by measuring the CV. So, from this graph we can understand when you are using this bare electrode, you can see that there is a baseline electrochemical activity, and when you are using this functionalized graphene coated electrode, there is a significant increase in the current, reflecting the enhanced electron transfer kinetics. And when we are immobilizing the anti-CEA antibody on this F graphene electrode, there is a slight reduction in the current due to the insulating nature of antibodies. And when you are using the BSA as a blocking agent, you can see that there is a further reduction in the current. It is indicating the successful blocking of non-specific sites. In today's practical demonstration, we will learn this more in detail. Before we learn about immunosensor biosensing study, let us briefly learn about differential pulse voltammetry, that is DPV. So, DPV is an advanced electrochemical technique widely used in biosensor studies for its high sensitivity and low detection limit. Compared to this CV, DPV applies a series of pulses over a linear potential ramp. The current is measured just before and after each pulse, allowing for the suppression of background currents and highlighting the Faradic process associated with the analyte. Let us see the biosensing ability of the fabricated immunosensor. So, once you modify the working electrode with the antibody specific for CEA, then you can incubate the working electrode with CEA antigen with a different concentration. And you can see here with respect to the concentration, the current goes down. If there is no antigen, so that is at 0 nanogram per ml, you can see here the baseline current corresponds to the unbound immunosensor. And you can see with respect to the increasing in the concentration of CEA antigen, the current is going down. And by using this technique, we can quantify even up to 10 femtograms per ml. So, that is the advantage when we are able to detect at the very low quantity. So, this biosensor can be useful for early diagnosis of disease. For in this case, we are using this CEA, that is a carcinoembryogenic antigen. So, this can be useful for early detection of cancer. So, this relationship between the current and concentration demonstrates the sensor's ability to quantitatively detect the CEA levels with high sensitivity. I hope you got the overall idea about the electrochemical nanobiosensor. Let us go to the lab and learn this technique more in detail. To fabricate an electrochemical biosensor platform, we need CEA antigen and its monoclonal antibodies. EDC, NHS, BSA, PBS, amine graphene powder, potassium ferrocyanide and potassium ferricyanide. This is a portable potentiostat from Metrome, which is connected to a laptop and is controlled by a software. The potentiostat has a connector for the screen printed electrode. This is a screen printed electrode which has a counter, reference and working electrodes. The middle portion is the working area where we have to deposit the transducer material. First, we will weigh 20 mg of amine graphene powder . Add this to 20 ml of 70% ethanol to make 1 mg per ml solution. We will sonicate the solution for 15 minutes for proper dispersion of graphene. You can see that the solution is homogeneous properly. Now, we will drop cast the solution on the area of the electrode. The electrode is covered with a film to only expose the working area. Drop 5 microliter of the material on the working area. And dry this electrode overnight in an oven at 37 degrees Celsius. You can see that the electrode has been modified using amine graphene. For immobilization of the antibodies, we need to first activate the antibody using EDC-NHS chemistry. For that, we require 50 microgram per ml stock of antibodies, 4 molar EDC and 1 molar NHS. First, add 10 microliters of antibody to an empty vial. Then add 5 microliter of EDC. And 5 microliter of NHS to make a ratio of 2:1:1. We will keep this for incubation at 4 degrees Celsius for 45 minutes. After incubation, the antibodies are now activated. We will drop 5 microliters of the activated antibodies to the 3 modified electrodes. Electrodes are then kept in a humidified chamber and place it in the fridge for 1 hour at 4 degrees Celsius. After immobilization, we will move on to the washing step. PBS is used to wash the electrode surface. Washing is done to remove the unbound antibodies. After washing, we will block the surface with 2% BSA solution. Take 5 microliters of BSA, add it on the working area of the modified electrode. Again, keep this in the humidified chamber for 2 hours incubation. After blocking, we need to wash off the excess BSA with PBS. Now, using cyclic voltammetry studies, we will compare the current response of modified and unmodified electrodes. For this, we need a redox coupler solution which is made up of 5 millimolar potassium ferricyanide and 5 millimolar of potassium ferrocyanide dissolved in PBS. The parameters of CV, that is cyclic voltammetry, have been applied in the software. Here we have the unmodified skin printed electrode, skin printed electrode modified with amine graphene, and modified skin printed electrode with antibody immobilized onto it. First, connect the unmodified electrode and add 50 microliters of redox solution to the working area. Run the CV. You can see the graph on the screen. Similarly, we will check the CV of modified skin printed electrode. You can see that in the modified electrode there is an increase in the peak current. This is a modified skin printed electrode with antibodies and further blocked with BSA. Here you can see a potential shift in the graph, which is due to the antibody immobilization on the skin printed electrode. Here is a comparison of the CV of the 3 skin printed electrode. Next, we move on to the electrochemical response study of the electrodes. For this, we will do differential pulse voltammetry. First, we will take the response of the modified electrode without the antigen. You can see the response on the screen. Now let us see the response of the electrode when we add the antigen onto the electrode. We will incubate the CA antigen on the electrode for about 10 minutes. Add 10 microdose of the antigen on the surface and incubate for 10 minutes. Wash the unbound antigen using PBS. Add 50 microliter of redox solution to the surface and take the response. You can see there is a clear decrease in the current when the electrode is presented with the antigen, denoting an immune complex formation. You can do this analysis further by incubating different electrodes with various concentration of antigen and recording the response of the sensor. As a summary, in today's lecture, we learned about what is biosensor, what are the various components of biosensor, and we also learned how does electrochemical biosensor work, both theoretically as well as practically through demonstration. Thank you for your kind attention. I will see you in another interesting lecture.